Good afternoon. Thank you for the invitation to come today. So I feel very fortunate to follow, I think, Tim and Tim, the two Tims, and Ian from this morning who really, I think, set the stage, a sense, for what I'm going to talk about, but I'm going to take you back much more into the preclinical setting and really focus on work from my team, trying to understand in a very basic research setting the molecular determinants of drug response in cancer. With the ultimate aim that what we're trying to do is facilitate the development of new oncology drugs and allow enhanced patient stratification. So I think it's fair to say there's been a really, I think, very disruptive changes in the field of cancer and cancer research over the last few years. And these have become from, I think, two very important developments. One has been due to the very large-scale sequencing of patients' tumors, much like Tim has just described, that's going to be done by Genome England. And to date, literally thousands of patients' tumors have been sequenced. Not all of them are whole genomes, but many exomes certainly, and many thousands of tumors from different cancer subtypes. And from that, I just really wanted to highlight a few, I think, key points that are important for what I'm going to discuss later on. The first is that these are identifying many genes that are causally implicated in cancer, what we tend to call cancer driver genes. And to date, there's at least 250 of what I would define as high confidence genes that we think with a high confidence are causally implicated in cancer. But we are discovering new genes all the time, particularly as new tumor types are sequenced and as larger cohorts of patients are sequenced. And we now know, or at least we've really confirmed what was established to some extent, that there are multiple cancer genes within each and every tumor, and that actually the constellation of different genetic changes varies considerably by each tumor type. And so what you begin to get is the emergence of actually what's quite a complex picture where individual cancers are very diverse. And so, as I think Tim said earlier this morning, in essence, there's huge diversity within the different tumor types and different patient populations. The second key development has been an emphasis on genetically targeted cancer therapies, which is, I think, in essence, what we're talking about here, based around patient stratification. And just briefly to say, traditionally, clinical trials have been done on large cohorts of unselected patients. And unfortunately, this has led generally to very, very low success rates. But increasingly, there's a recognition that if one can stratify patients appropriately, you increase the probability of observing clinical responses and ultimately success. And really, it's based on this identification of these biomarkers that certainly can help with this process. And I'm going to use this word a lot, biomarker, and what I mean by that is really that's any molecular change within a cell that could help identify or stratify patients. So this could be a mutation or a gene expression signature, for example. So let me just go back. So, sorry, one thing I think it is worth mentioning is that identifying these biomarkers, excuse me, remains very difficult. And there's many reasons for that, but certainly one of the key reasons is due just to this genetic complexity that we observe across different cancers that makes this stratification very, very difficult. One very nice example of this type of genetically targeted therapy is the development of ALK inhibitors for a form of non-small cell lung cancer associated with this very specific gene fusion. So this represents a relatively rare subset of patients. It's less than 5% of these lung cancer patients who have this particular molecular lesion. And these patients respond very well to an inhibitor of ALK kinase called crizotinib. And that's just shown here where you have a tumor and you see a very significant response following therapy. Now, it's important to point out that patients that don't have this particular molecular event do not respond to this particular therapy. And in general, you get about a 60% response rates. So again, even though we have a biomarker, it doesn't necessarily explain the full range of responses. And this leads to about a 9-month 9-month progression-free survival overall, which is better than the previous standard of care. The reason I really highlight this is that it only took 4 years from the initial identification of this fusion to clinical approval of crizotinib, which in drug development terms is extremely rapid. And it really goes to show, I think, where you have a clearly stratified patient population with a good understanding of the molecular biology that underpins the disease, you can rapidly translate things into the clinic. However, I will mention, and it has been alluded to before, unfortunately, these patients do acquire resistance to this therapy, and this appears to be a feature of the majority of targeted therapies. And I'll come back to this a little bit more later on. I actually did a survey of oncology drugs in September of last year, looking at the website of the National Cancer Institute in the US, as well as the FDA. And there's actually 67 molecularly targeted oncology drugs. And you can see the approvals per year, so you can see it's fairly consistent. And these actually represent about 30 unique molecular targets in oncology. And just under 40%, or sorry, just over 40% of these are actually linked to a biomarker that allows us to select patients in the clinic. So already there has been a significant adoption of patient stratification in the oncology setting. And indeed, there's a number of new therapies coming through the pharmaceutical industry's pipelines. That's depicted here, where what you have here is the year and the number of compounds in clinical development. And the key one here is oncology. You can see that there's literally hundreds of molecules coming through this drug development pipeline. And of course, this represents an enormous opportunity, but it also represents an enormous challenge, because most of these compounds will not have a biomarker that will allow us to identify the patient population that we think would be most responsive to a particular therapy. So there's a need for many of these drugs to really identify those biomarkers. And in fact, overall, historically, in oncology, the success rate for oncology drugs is extremely poor. So most drugs fail. They fail in phase 2 clinical trials, and they fail because of lack of efficacy. And this is a very expensive process. There's a lot of waste of money, of patients, and so we're really seeking to kind of improve this process. And of course, this is only getting more challenging as we develop new drugs to more diverse sets of targets. So we're moving out of the world of just targeting kinases. We're targeting the metabolic machinery, the epigenetic machinery. And it's very unclear how we're going to begin to position those molecules in the clinical setting to maximize their efficacy. So what we've been doing over the last 5 years or so is really just trying to bring together this wealth of genetic information that is now available together with the drug development pipelines of the industry, academia, and the biotech sector. And what we do is very, very simple. We take cancer cell models and we perform differential drug sensitivity screens. And then we link that drug response with genetic information that we have for those models. And this has a number of different applications. So first is it allows us to understand those molecular features that actually determine response from a very fundamental type of way. Of course, this also allows us to think about developing biomarkers for new therapies as well as to improve existing therapies. There's also opportunities to repurpose existing drugs into new indications. And in some cases, we actually gain new insights into mechanisms of drug action, particularly where, for example, we might repurpose a drug. And so there's really, I think, a number of very useful bits of information one can gain from this type of experimental design. The assay we use, again, I think is quite simple. We take collections of drugs. These are not vast chemical libraries. They're very carefully selected molecules. and we screen them. We take one cell line in a plate, and we then screen it with these molecules. We generate a dose-response curve for each cell line to each drug, and we measure the relative sensitivity of that cell model. And the way we do that is generally we record a value, either the IC50, which is the half maximal inhibitory concentration, or other metrics like the area under the curve. And again, these just really try and quantify the relative sensitivity of each cancer cell model. We usually treat the cells with drugs for either 3 or 6 days. We do a very simple fluorescence viability assay at the end of the experiment, and then we use various statistical methods to link this drug data with different types of genetic data. And really the workhorse of this effort to date has been based around cancer cell lines. And I think it's fair to say that these are really the workhorse of much of cancer research over the last almost 50 years now. And this is just a picture of Henrietta Lacks, from which her tumor we derived the HeLa cell line, which is, I would say, very famous, if not infamous. But nonetheless, it has been used ubiquitously in research. There are over 70,000 citations in the literature with the word HeLa. And despite important caveats, cancer cell lines have really provided very fundamental insights into our understanding of both normal and cancer biology. So here at the Sanger, we have a collection of over 1,000 human cancer cell lines. That's depicted here. They're classified according to the tumor subtype which they represent. We're using the TCGA classifications here. So for example, we have 52 colorectal adenocarcinoma cell lines. We have 51 breast cancer cell lines, and so forth. And we have this very large collection, or as large as possible, to encompass as much of that genetic diversity of cancer as we possibly can. And I would actually argue even this collection is inadequate to really encompass that diversity. Another key point is that these cell lines have been very deeply molecularly characterized. So what does that mean? That means that over 1,000 of the cell lines have been whole exome sequenced. We have SNP6 copy number for over 1,000. We also have array-based and increasingly RNA sequencing-based expression data. We also have genome-wide methylation data. And as I mentioned, we have drug response data for many of these lines. And altogether, across these 5 data sets, we have now just under 1,000 cell lines that have been characterized to this level. And what this does is it enables us to look very deeply for those molecular features or combination of molecular features that are most predictive of response to this set of drugs. The drugs themselves, as I mentioned, are very carefully selected to target key cancer pathways and processes. That's just depicted here by different hallmarks. So for example, we have a relatively large number of tyrosine kinase inhibitors. These are key proteins involved in cancer, and also a major drug target from the pharmaceutical industry. But we're very interested in exploring very novel target space as well, looking at things like cellular differentiation, for example. The compounds themselves are sourced commercially, so we buy them, but we also work very extensively with the pharmaceutical industry, academia, and biotech to bring these compounds into our screening platform. So to date, we've screened about just over 600 compounds across the majority of this cell line collection. So this is 1,000 cell lines, and where it's kind of brown, we have data. We've generated over 600,000 IC50 values. And this has involved screening about 200,000 384 well plates. So it's a very large data set, as I mentioned, very deep and rich, both in terms of the amount of genetic data, but also the amount of pharmacological data. And then we can ask very simple questions. So for example, in this particular here, we've just said, in response to this particular drug combination, so this is an inhibitor of 2 kinases, BRAF and MEK, does the presence of this particular mutation in a gene called BRAF, which was introduced by Tim this morning, does that lead to a differential sensitivity to this particular combination of drugs? So what you have here is each circle represents the IC50 of an individual cell line, and the color represents the tissue type from which it was derived. And hopefully what you can see is that on average, if you have a BRAF mutation, the cells are more sensitive to this particular combination of drugs. So very simple observation, and in fact, this particular combination, which is dabrafenib and trametinib, is now clinically approved for the treatment of melanoma patients who have this particular genetic lesion. So you can capture these types of things using these very unbiased screening efforts. This is just one example, but what we endeavor to do is to look for these types of relationships, not just for one drug and one gene, but across hundreds of drugs and hundreds of genes. And to do that, we use a number of statistical methods. Don't worry, I'm not going to go into any details about them, but just to say, we use these to really fully interrogate these data sets. So for example, we've taken sets of driver genes that I referred to earlier. This is a set of 338 genes, as well as amplified and deleted genes. And then we use statistical methods to link this with our drug data. Our method of our method of choice is this analysis of variance to do that. But we have other methods, including things like logic models and elastic net, which are basically different approaches that add different layers of information and enhance our modeling. But I'm not going to go into a lot of detail, and I'll just give you a little flavor of what this type of data looks like. So we depict this data using what's called a volcano plot. I'm afraid I don't think it's projected particularly well, but what you have here is each circle on this plot, I'll start up here, this big circle represents the interaction between an individual drug and gene. And in this analysis, we probed 145,000 different interactions. We identified about 1,100 that are statistically significant, and a subset of those have a very big effect on the sensitivity to a particular drug. So these are gene-drug interactions. So for example, this particular association is associated with resistance, and it says if you have a mutation in P53, the tumor suppressor, you are predicted to be resistant to a drug that targets MDM2. Okay? This is actually a well-established relationship in the literature in terms of the way these drugs work. Another example, again, here we've shown that if you have a mutation in BRAF, you would be predicted to be sensitive to this BRAF inhibitor, dabrafenib. Again, something that's observed clinically. And I'll just highlight one other association, which is this association between this particular gene fusion, EWS-FLI1, which is characteristic of a childhood bone cancer called Ewing sarcoma. And this analysis predicted that you would be those cells are sensitive to a PARP inhibitor. And so using this type of analysis, you really identify thousands of these interactions. You can then dig a little bit deeper into these, and you can ask very specific questions like, what drugs would be predicted to be effective in the context of cells that have an amplification in ERB2? So this is also known as HER2, as commonly observed in breast cancers. And what you find is that through this unbiased screening approach, we pulled out a number of different drugs that target EGFR and ERB2. So again, this is largely what's expected, but in this very unbiased way, we're able to pull out many different drugs that target this. We can then dig a little bit deeper and try and understand the data that underpins that. And what we see, for example, is we have a small number of lines that have this amplification, and they're very, very sensitive to this particular molecule. In contrast, we had a number of drugs targeting MEK kinases that were suggesting that these cells would be resistant to a MEK inhibitor. And again, that's just shown here. So you can kind of drill down into these data and get a much more detailed picture of what's going on. We can find these known things, but obviously we're interested in finding novel associations that can help improve the development of these particular molecules. And I'll just highlight one particular example that has come out of this screening effort. This was that relationship I showed you between sensitivity to olaparib in the presence of the EWS Fly1 translocation. We followed up that association, and it's been confirmed by several other groups. And on the back of that work, that has led to a number of clinical trials, including this one here, evaluating the use of olaparib in combination with a chemotherapeutic in those patients. And so this is just an example of how we can take results from the cell line screening effort and try and transition those into development of new cancer therapies. There's been some discussion today about the use of combination therapies. There's a recognition now that for most molecularly targeted agents, resistance is a major issue, and most patients relapse in under a year. And there's a number of reasons for that. So for example, here, you can get immediate feedback signaling that attenuates response. Tim introduced this earlier. This is in colorectal cancer. In patients that have this BRAF mutation, if you inhibit vemurafenib, you would expect to turn off this survival pathway. But what actually you do is you release a negative feedback loop that allows those cells to survive. And so what you need to do to get an effective therapy is to combine an EGFR inhibitor together with a RAF inhibitor, and increasingly now with a MEK inhibitor as well. So this is a feedback activity that bypasses the activity of this single agent. Another example of where combinations are thought to be effective is with bypass tracks. So you have, for example, two receptors here that signal through this pathway, and you really need to hit both of them to turn off that pathway. We also observe acquired resistance, and it's thought by combining agents, you might be able to delay the acquisition of resistance. And there's also hope by using combinations that we can identify synthetic lethal interactions where the presence of one drug induces sensitivity to a second molecule. And so we feel that our screening platform is very useful for this type of observation. The number of combinations to explore is extremely large, and this type of high throughput approach in cancer cell lines is very amenable to trying to take on this challenge. So over the next few years, we are performing very large-scale combinatorial screens in these cancer cell models. The idea is to do what we would call an anchored screening approach. We're taking 20 clinically used drugs. These are what we call the anchors, and we're screening them in combination with 250 more experimental molecules to give approximately 5,000 two-drug combinations. We're going to do this using what I would define as an ultra-high throughput screen across 1,000 cell lines. This will require us to generate 50,000 1536 well plates and 50 million data points. So the scale of effort here is very large to really get a handle on the scale, the number of combinations, and the number of different cancer subtypes. And ultimately what we'll do is we'll model these drug interactions and use those type of statistical methods that I've already introduced to try and understand what are the molecular features that would predict sensitivity to different two-drug combinations. And of course, we're using these clinical anchors because the feeling is that this will enhance our ability to transition these findings into the clinic where you're pairing with something that's already clinically used. The other advantage is that interpretation of these combinatorial effects can often be quite difficult, understanding why certain combinations work. And we felt pragmatically by having one molecule where we understand the activity, at least to some extent, will facilitate the interpretation of these data sets. So we've initiated this project just in the last few months. And just to give you a flavor of the type of data that we generate, this is some data screening a variety of drugs together with a PI3 kinase inhibitor, depicted here. And what you have, sorry, is this working? Yeah. In black is the response to a PI3 kinase inhibitor as the dose is increased. Sorry, to erlotinib, which is an EGFR inhibitor, sorry. The red line is the additive effect if you add in a PI3 kinase inhibitor. And the green line is the actual observed sensitivity in that experiment. And so what this is telling us is that there's a very great synergy here between the presence of an EGFR and a PI3 kinase inhibitor in this particular cell line. And so now what we need to do is see if there are other cell lines that respond to this particular combination, and if so, what are the molecular features of those models that potentially underpin that sensitivity? So I've talked a lot about cell lines, and as I've said, we feel like they're an enormously useful model, but they do have a number of drawbacks that I think many people in this room will be aware of. I've just highlighted two here. One is that it's quite clear that this currently available set just really doesn't capture the diversity of tumors that we know exist in patient populations. So for example, some tumor types, we just really don't have a very large collection of models, prostate being the classic example. And even when within a model where we have lots of tumors, we're not capturing all those driver genes, all those molecular changes within this set of models. And of course, these models have adapted in culture, and so don't necessarily, again, give a fair representation of the clinical prevalence of different populations. So over the last few years, there's been some really exciting developments in the way that we can culture cancer cells in the laboratory. And this is really opening up, I think, a whole new window of opportunity. And one of these approaches has been the development of what's referred to as organoid culture. And what this allows you to do is to take tissue from patients, both normal and malignant tissue, and grow it in the lab. And you can go from a single stem cell right up to a differentiated three-dimensional culture that recapitulates many more of the features of the tumor of origin. So for example, it has crudely captures some of the architecture of that organ, as well as the cell type. So you get differentiation of these stem cells into the different cell types. You're able to do this now from a host of different tissue types, so it's very applicable across many tissues and tumor types. It requires specialized 3D growth conditions. You can grow them for a long time, and a key point here is you can derive these at a very high efficiency from patients. And what that means is that you can begin to sample all those patient populations. So there's not a huge selection bias that is a real problem in our current culture model. So we can begin to develop these models we need that reflect different patient populations. So over the last couple years, we've had a very fruitful collaboration with the group of Hans Clevers and Matthew Meyerson to see if we could build what we would call a proof of concept biobank of these models for predictive modeling. And the idea is as follows. We took 20 patients and we received surgical resections from those patients. And from those, we were able to derive organoids, both from the tumor as well as from adjacent normal tissue, and grow those in the lab. We could do this with a very high success rate, so over a 90% success rate doing this. And from these samples, we did DNA sequencing analysis, we did our RNA expression analysis, and we did drug screening. In addition to that, we also always acquire a biopsy reference sample so we can compare the tumor to the original biopsy from which it was derived to see how faithfully it represents the tumor of origin. And I'm going to just summarize a lot of data in just one or two slides here. And the key point is that these cultures seem to be doing a very good job of capturing key aspects of colorectal cancer biology. So for example, these are the mutations that are observed in known cancer genes in our colorectal organoids. So just here, first of all, as Tim introduced this morning, we can clearly define a hypermutated and a non-hypermutated population based on the total mutational load. We see that the most frequently mutated gene in this cohort is APC, and you can compare that In our organoids, it's 85% of samples have an APC mutation, and this closely correlates with what's observed in clinical data sets of hundreds of patients. So the frequency is maintained. And a similar story for other known cancer driver genes in colorectal cancer. So for example, PIK3CA here. So we're capturing the major driver genes, and we're capturing them at a frequency which is broadly similar to what we observe in patient cohorts. And again, just to summarize additional data that I don't have time to go into today, but these organoids retain the histological features of the tumor of origin. They also retain features of the heterogeneity and clonal architecture. So many of these tumors will have multiple subclones, and this clonal architecture is actually maintained in these models. And as well, we are using gene expression analysis, we can identify these different molecular subtypes that were introduced again by Tim this morning. So in aggregate, it does seem like these models are doing actually quite a good job of really capturing the biology and the hallmarks of these colorectal tumors. We have then taken these different models through a high-throughput drug screen. And just to give you a little bit of a flavor, for example, we did 20 models through 85 different drugs. The drugs are selected, again, to include those that are used clinically for colorectal cancer, other chemotherapeutics, as well as preclinical and experimental compounds. And I'm just going to show you one result, which is response to cetuximab, which is clinically approved for the treatment of colorectal cancer patients. This is an EGFR inhibitor that's given to patients who do not have a mutation in KRAS. And what you can clearly see here, hopefully, is that the sensitivity in the setting of wild-type KRAS is greater than in the setting of those that have a KRAS alteration. What's interesting as well is that we can see, sorry, this laser pointer is not working, but there's 3 circles at the top in the KRAS wild-type. So these ones are insensitive to cetuximab, even though they're KRAS wild-type. What's interesting is that one of these has a BRAF mutation, which is a known mediator of resistance to this particular therapy. And of course, there's now an opportunity to begin to investigate these models to understand perhaps why the other 2 are not responding. So this is where we see some of the power of this particular approach. And so we're very excited. We think there's now an opportunity to begin to develop a new set of cancer models using these technologies that really overcome so many of the limitations of our current models. So we can begin to acquire samples from both biopsy samples and surgical resections, and develop models across potentially thousands of matched tumor normals from both common and rare tumors. And there's also opportunity to integrate these into different clinical trials to really try and understand the variation in patient responses that are observed in those trials. We would suggest that these should be highly annotated, so they would be linked to clinical data, have a documented sample history, and be derived in a very standardized way, subjected to in-depth genetic characterization and drug profiling, and to build a biobank of these. And we think these will have a very transformative effect. They'll have multiple applications, for example, modeling drug response, exploring tumor heterogeneity, modeling drug resistance, comparing primary versus metastatic disease, looking at therapies that target stem cells, and so forth. This technology is really opening up a whole world of opportunities to really ultimately improve patient therapies. So just to summarize briefly, we believe that these pharmacological screens really have the capacity to identify clinically relevant biomarkers. There's also applications in really trying to combat the problem of drug resistance. And I didn't go into it, but there's also opportunities using genetic screens to identify new oncology targets. And ultimately, looking forward, a new generation of cancer models which really better capture the biology of tumors, I think are going to open up a whole new opportunity for improved predictive modeling. So with that, I'd just like to conclude by thanking the people involved. It's very much a team effort, and I'd like to thank you, and I'm happy to take any questions.
Thank you very much for an excellent talk. We've got time for one question.
Sorry.
Thank you. I'm just wondering if you can tell us, you mentioned it just at the end. You know a lot about this, and you've got to sell it pretty hard. But what can you do about the co-evolution, which is what cancer is? You mentioned metastasis at the end.
Yeah.
It's a very tough problem, I recognise, but there's a co-evolution, isn't there, with the subject involving those metabolic genes in which you didn't have too many drugs.
Yeah. So there's co-evolution in multiple ways. So there's co-evolution within the tumor subclones, which these models would allow us to explore. This model system is restrained in the sense of how it can explore host interactions. Yeah, so this is primarily around cell autonomous events. Of course, one can think about more complex co-culture systems and so forth, where you can begin to model those things. But I think certainly the emphasis at the moment is more on those cell autonomous events and perhaps that dynamic of different clones within a tumor.
A single patient, and in continuity, you were able to biopsy, if you will, their metastases. There are diseases which are very obliging in terms of metastases popping up in accessible places.
Yes.
Horrible though they are. You would get tremendous power of evolution, as it were, in an individual that could be shared amongst a few individuals, and you'd know a common core.
Yes, I think that is the opportunity. You could do serial biopsying over the course of treatment. You could do multiple metastases to look at whether there was some kind of clonal evolution. I think the opportunities are enormous using these types of technologies.